Hello, my name is Rong. I'm an assistant professor in the Department of Biochemistry and Molecular Medicine. Welcome to WVU. My lab is very interested in research on molecular motors. Molecular motors are essentially the engines of life. They function very similar like the engines in our cars to convert chemical energy into mechanical motion. There are a wide variety of motor proteins, and they power different kinds of motions from muscle contraction to cell division, and they also transport different cargoes from one place to another. Motor proteins also build cellular structures. One of the structures we are very interested in is the stereocilia of auditory hair cells. They are clusters of surface protrusions filled with motors and cell-skeletal filaments. And they organize themselves in an incredibly precise pattern like a stack of organ pipes. This exquisite morphology allows stereocilia to translate sound vibration into electrical signals, which is a process essential for our ability to hear. Our long-term goal is to understand how motor proteins participate in the formation and mechanotransduction of the stereocilia, and how their defects lead to deafness and many other diseases. Because we are interested in motors and cell-skeletal dynamics, live cell microscopy is central to our research. We can label different proteins with fluorophores and study how they interact with each other in space and time. This could be very informative in understanding the dynamics of the cell skeleton. Another powerful approach to study molecular motors is to take them out of the cell and to recapitulate their behaviors in a reconstituted system. This allows us to identify the minimal sets of proteins required for the function, and also it's very useful in studying mechanisms that sometimes may not be accessible in the complex cellular environments. The process of taking something apart and putting them back together can be very helpful in understanding how something works. Students in my lab will be exposed to a range of interdisciplinary approaches, from eukaryotic protein purification biochemistry to quantitative imaging and single molecule biophysics. I encourage students to ask fearless questions, to think independently, and also to attend national meetings to start to develop your professional network early on. Students are my first priorities, and my goal is to help you succeed and to achieve your career aspiration after graduate from the PhD program. I look forward to meeting you all.